So big picture for a moment here. So we study cardiovascular disease. It is a leading cause of death. 17 million deaths per year globally, according to the World Health Organization. 2 million people in Canada, or about 7% of our population, reports living with cardiovascular disease. About 66,000 people die per year in Canada from cardiovascular disease, and that's about one person every 7 minutes. In terms of economic costs, it's about $21 billion per year in physician services, hospital costs, lost wages and productivity, and about a log order in magnitude greater in the United States and the UK, where there's more people. So you're looking at outrageous numbers like $200 billion per year. The University of Guelph invests in science from basic research to translation and outcomes, including our mandate and our center, which again is dedicated to discovering novel ways to diagnose heart disease, advance treatment, and train the next generation of clinicians and scientists. So that being said, it is my absolute pleasure and honor to introduce our first and keynote speaker, Dr. Lorrie Kirshenbaum. I had to make slides because he has just done such an enormous amount of work, but I've tried to summarize here some of the areas of research that he does. He is truly a pioneer in the molecular mechanisms and signal factors that govern cardiac gene expression during early cardiac growth and cell cycle and cell death. He also works in cell cycle control and cardiac regeneration, and this is not just basic science. These have far-reaching implications for patients with heart disease and then reach into other areas of health and disease as well, especially cancer. He's also opened up new areas in caspases and mitochondrial defects, and indeed he brought a whole new player into the field recently, cloning the novel and key regulatory mitochondrial death factor BNIP3, which he'll probably talk about today. In terms of his accomplishments, Dr. Kirschenbaum is a Tier 1 Canada Research Chair in Molecular Cardiology and previously Tier 2. He is Director of Research Development, Faculty of Health Sciences, University of Manitoba. He's the Acting Director of Basic Medical Research at St. Boniface Hospital Research Center. He's Professor in the Departments of Physiology, Pathophysiology, and Cardiovascular Sciences at the University of Manitoba, and cross-appointed to the Department of Biology at San Diego State University. He did his training initially in Canada, and then he went to the States to do a postdoc at Baylor College of Medicine before coming back to Canada. He has won, and I couldn't figure out which ones to pick, so I just wrote numerous awards for innovation and research. He's invited to give talks around the world, both as a speaker and as a session chair at the major meetings. And he reads grants for organizations like CIHR, Heart and Stroke, sometimes as a reviewer, sometimes as a consultant, and also the prestigious committee chair. Okay, but one of the things that I like most about Lorrie that really strikes me is that he leads by example, not just through the research that he does, but also through his interactions with the students and faculty and his support. And I'm very excited that he'll get a chance to meet some of you people today through the poster session as well. And so, Lorrie, I have a plaque to present to you as our keynote speaker for Cardiovascular Research Day from the Center for Cardiovascular Investigation.
Thank you so much. That's terrific. Thank you.
In the box.
Thank you.
Okay? So I'm going to give you this.
Okay.
This is for the video.
Video.
And then this is the audio.
Okay. Perfect.
And you know how to swap this?
You want to switch it over?
Yep. You can switch it over.
Okay. Perfect. Thanks.
It's just a piece. Just escape out of here. Yeah.
Just pick anyone.
Yeah. You can give Zam's. That'll be good. That's the
Okay. Let me just get this figured out. Kim, it's the top button, yes?
Yeah.
No, done. I'll use the mouse. I think the battery might be toast, but anyway, that's okay. Was it me? Which one? Yeah, that's what I was pushing. Oh.
Oh, actually, it's just below.
Just below. Okay, good. We're in operation. Well, first of all, let me express my sincere gratitude to Tami and the organizing committee. I mean, I've known Tami for many, many years, and I'm absolutely thrilled, delighted. I can't express my gratitude to Tami and the rest of her crew to be here. The institution is absolutely fantastic. I've met so many great people, some old guard and new. First time to Guelph, and what a great institution. One of the interests I have, of course, is meeting people along the way, and you never know who you're going to run into. And several years ago, Tami and I sort of commensurated at dinner, her first trip to Winnipeg, I think, as a graduate student or postdoc. We had a meeting, a young investigator forum, and there she was at her poster telling us about the circadian rhythm. And it had to be one of the very first demonstrations that circadian was important in cardiovascular health. And as a pioneer in the field, she was basically, just trailblazing forward. It was tremendous. And then fast forward, almost 20 years later, and here's Tami running a program, and as from 4 to 15 or so of you as faculty is terrific. So I'm honored to lead off. I'm delighted to speak with my colleagues this afternoon, and what a tremendous day in celebrating research. So what I would like to share with you, I changed the talk title, How to Mend a Broken Heart, and what I'd like to tell you a little bit about is how our research applies clinically, and take you from a clinical problem, 2 different clinical problems, on how we led to our research program on BNIP3 and how it might be important for treating cardiac failure in the next 5 to 10 years. So with that said, I'll take you on a very interesting story. So people like case histories. So this is a true story. This is a case history. A 72-year-old lady shows up with chest pain at the hospital, and this is early morning. So there is a circadian component to this. I realized this after without actually putting the talk together. So she shows up early in the morning, and as Tami will tell you, that's typically when this happens. She has no radiating pain, classical heart attack type symptoms. She's ambulatory, no previous cardiac symptoms. She has hypertension, hyperlipidemia. She's not a smoker, non-diabetic. The medication she's on is a beta blocker, metoprolol, some ACE inhibitor, and some pravastatin to help her lipids. When you examined her, she had, reasonable blood pressure, 106 on 61, regular heart rate, no real, anything impressive upon examination, no JVPDs, the jugular venous depression, or normal carotids, et cetera, et cetera. So, what's going on with this woman? And, neurologically, she's normal, so she's not, hallucinating, no evidence of stroke. You looked at her ECG, and just to bring, this wasn't going to be a clinical talk or bring you into clinical cardiology. Tim will look at this, and he'll pretty much look at and tell me he could read this. But the point I just want to show you is evidence of an ST segment elevation in these leads. These are the precoital leads, V1, V2, and V3. And so she had some evidence of a previous heart attack and some evidence of cardiac dysfunction. And when we cathed her, and you take a look at this individual, you can see that there's some pleural effusions and some dilatation of the heart, and there's so evidence of some heart failure, some congestion. And so my colleague basically sticks a wire in her and takes a look, doing an angiogram. And you can see, in contrast to the right coronary artery, this is the left coronary artery, this is the left coronary artery, left anterior descending. You can see there's an occlusion. So if this was a normal vessel and it was opened, this would be highly perfused, and essentially the heart would function. But there's no blood flow here because there's an occlusion, and consequently that shows up on the angiogram as a blockage. So then my friend puts a stent in and basically opens up the vessel, and she gets great flow, and you can see that this is now a post-stent view. The images are a little bit different in terms of how we took them, but essentially you can see that flow is regained after stenting. Okay, so that's all good. She had a blockage, we restented her, and this was what was really interesting. Her ejection fraction a few weeks afterward, this is 2 weeks afterward, is only 35%. Despite the fact that she had great flow. I mean, the vessel looked patent, but yet her ejection fraction was quite poor. And I'm just going to play that again, if I can. Whoops, I'll go back, if it'll play. Let me just click on this. Whoops, just click on this. Where's the mouse? I can just click on this. What's really impressive about this is that you can see that when I play the video again, I want to draw your attention in this long axis view to the left ventricle. You can see that it's basically baggy and akinetic. It's not doing anything. And so basically, she's basically in heart failure. You can see this portion of the heart is not moving. And obviously that's why her ejection fraction is so low. So despite coronary flow, reperfusion, she has sustained reperfusion injury. And this is really what I'd like to talk about, and how cell death involves, reperfusion injury and cardiac remodeling. So the good news here was the patient walked out of the hospital. She had this large MI, a stormy visit to the hospital, a little bit of a stay. But the problem is now she's going to develop heart failure. And it's predicted that individuals, even with coronary flow post-MI, will develop heart failure, usually 2 to 5 years post-stenting or post-MI. So this is very common. You see this with large, I should point it this way, large infarcts. Maybe I should use the mouse. It'll, I think, show up a little bit better. I'll do this. Maybe I should stand over here. So essentially, this is really quite common. The clinicians will verify that this is something that happens. And so the question is, we reperfuse this lady, and there's no question that reperfusion is important, bringing the oxygen back into the cells, preserving cardiac function. But the double-edged sword is it's causing cells to die. And so this shown here is a really interesting statistic that was taken many years ago from Brunwald and Mark Pfeffer. And this was published in 1990 and some early work in the New England Journal in 1988. And we can see clearly this is 3 weeks, and this is 3 weeks to a year. The ventricle dilates. This is dilatation, and the person ultimately ends up in failure. And so the reintroduction of oxygen is really critical to save the cardiac cells, but the question is, how do you prevent them from dying? And so this has been the biggest issue in cardiac medicine for the past several years. So the idea is to develop an understanding of reperfusion injury, ischemic reperfusion injury, so that new therapies or treatments could be developed. So I just want to highlight, Tami kind of touched upon this, that myocardial infarction affects many people, 350,000 deaths. This was taken from a US slide, and more importantly, it exceeds cancer. And if you looked at the combination of how many deaths are following MI, you can see that it substantially accounts for almost 50% of cardiovascular related deaths. So this is a huge issue in North America, and it's getting worse, and largely due to ischemic heart disease. Okay, so the physiology is that we have a normal heart, presumably have an MI, this anterior infarct, and essentially the heart will compensate by cardiac remodeling and resulting in cardiac hypertrophy. And following the law of Laplace, the increased wall thickness basically offsets the defective heart, resulting in cardiac hypertrophy. So physiological hypertrophy, people can live for years with this. We see this in athletes, we see this in individuals, but for some reason in certain people, the heart basically fails. So you activate a variety of stress signals, this compensatory mechanism fails, and ultimately the person results in, develops heart failure. So the question is, what's going on in this remodeling process? How can we mitigate this event, and how do we prevent heart failure? So there are several things that are going on. Early days, we were looking at this and asked the question, since cardiac muscle cells don't proliferate, they're typically post-mitotic, despite the fact that there's some reparative mechanisms going on in the heart, some stem cells, etc. Those events are very, very meager, and they're insufficient to really improve cardiac function. And so the question is, what's happening post-MI? There's inflammation, there's progression of fibroblast infiltration, there's matrix remodeling. We'll hear about that a little bit later. But importantly, there are cells, cardiac cells, the muscle cells, that are stimulated to die, but they haven't quite yet completed the process. So the process of absence of oxygen will actually activate a cell death cascade that will initiate cell death, but it won't kill the cells. And so the issue is trying to slow the kinetics of percutaneous reperfusion when you put a stent in, or trying to know that if someone's coming in for bypass surgery, give them a drug that will delay the cells that were ultimately predisposed to die from dying would be ultimately a great therapeutic. And so many people over the years have been looking at this. And so the question becomes, can you treat this clinically? Can you prevent cardiac cell death occurring following MI? So let me tell you a little bit about the story. So pre-1980, no one really knew about cell death. They basically opened up a textbook, and there was a EM, or there was a H&E stain of some cardiac cells, and they were dead. And people believed it was predominantly necrotic, and it was a random act of cell death and cell suicide, and that was it. And then come along Sid Brenner and Bob Horowitz, and they, using the C. elegans model, were able to identify that deaths that do occur within the cell, and I did say deaths, so not just singular, are regulated. And that was ultimately the evolution of programmed cell death. So the original description was by Al Wiley and Kerr. These were pathologists who were looking at human T cells, and I'll show the images. And they basically identified these dysmorphic-looking cells by electron microscopy that were dead, but they didn't have the classical characteristics of necrosis. And Horowitz and company basically identified in C. elegans that there were 131 cells basically that are killed off sequentially during development, and if you interfere with those genetic pathways, the worm, the C. elegans, won't develop normally. And that was where the Nobel Prize was awarded a few years back, and why he's obviously, I'd be smiling too if I won a Nobel Prize. But anyway, in fact, the key here was the key observation that the deaths that were occurring was not just simply necrotic cell death. It was programmed, it was timed, it came on specifically, and more importantly, there were specific genes activated in this worm that were instructing these cells to die. So if you think from a developmental perspective, you know, the formation of our digits is really the regression of the tissue in the interdigital spaces. So when you look at a developing embryo and you see that baseball mitt, we're basically carving out the fingers because selectively the webbing disintegrates, not the fingers. So how does it know? How does the webbing know to disintegrate? So it's all genetically regulated, and that was really the birth of programmed cell death, or apoptosis, or programmed regulated form of cell death. And this is the electron microscopy I was telling you about that was really kind of interesting. Imagine guys are looking at T cells, and they found that these cells were really dysmorphic looking. So the apoptotic, classical apoptosis, is basically cell death. The membrane outside the cell is intact. The nuclear material is basically fragmented. You can see all this blebbing and, you know, disrupted cell. There's no question. ATP is still maintained in the cell for some time. But the key here is that the plasma membrane remains intact, and there's an absence of inflammation, usually in an apoptotic form of cell death. Necrosis, on the other hand, is really quite different. This is classic textbook necrosis. The cell would basically lyse, the intracellular contents would be dumped into the interstitial spaces, it would evoke inflammation, and post-MI, this is typically what happens. So the mode of cell death post-MI is largely necrotic, and it usually impacts cardiac cell death more so than apoptotic cell death. We know that now, but my lab was one of the first to show that apoptotic cell death does occur. More recently, we identified that necrosis is also programmed. So you have two forms of programmed cell death in addition to something called autophagy, which could also be a form of cell death or maladaptive in some cases and promote the demise of the cardiac cell. So we find later that apoptotic cell death, necrosis, and even autophagy have overlapping family members that actually determine under certain conditions what's going to happen. The cool thing about apoptosis, it's a stealth form of cell death, and as I mentioned, there's no inflammation. And this is basically involution of breast tissue after nursing. So here's, 10 days after lactation, day 2, day 6, day 9, and you can see that the alveolar spaces, the cells actually die off, but there's no inflammation. And this is cyclic, obviously. Post-MI, we see that there's damaged tissue, there's evidence of apoptotic cell death, and of course this is permanently injured, whereas in the breast tissue, we see that this is highly regulated. So the MI is to demonstrate the contrast that apoptotic cell death does occur, but in fact, it's a much different context. Here again, post-MI, this is an H&E staining taken from a colleague of mine, Hadamasa O. And so early necrosis, you can see on the border zone, there's necrotic cell death, there's also apoptotic cell death. And if you're not looking carefully for both, you can see whatever you want to see on an EM or an H&E staining. So I'm saying you have to be careful when you're doing the analysis. And what's more importantly, there's a spatial and temporal activation of these pathways because it isn't definitive that one pathway over another predominates. Although I did say, and it's true, that early necrosis is the predominant mode of cell death, and it's programmed. So it is indeed programmed post-MI. So let me take you through the signaling pathways that we've been engaged in over the past several years, what leads to cell death accordingly, both apoptotic and necrotic. So to remind folks, because I'm sure you have seen this before, the majority of cell death that was originally studied involved tumor necrosis factor alpha and some of the other death receptor activators such as FAS. And so what the classical model is that these ligands would activate a death receptor, the death receptor would then recruit to the cytoplasmic surface of the receptor various proteins. And the DISC stands for death-inducing complex. This would then recruit a number of other proteins, such as Tami had mentioned, this caspases. These are special enzymes, proteases, that become activated. And they go and activate downstream caspases, and the whole apoptotic cascade gets activated until the intracellular proteins get digested and ultimately leading in programmed cell death. And so the apoptosis was the key here. And what's shown here on the far right is the mitochondria. And so people studied the extrinsic pathway as just a simple death receptor pathway, and there were folks studying the intrinsic pathway, which now we believe involves obviously the mitochondria. And so defects in the mitochondria, both at the outer membrane and the inner membrane, ultimately lead to the demise of the cell through both an apoptotic mechanism and also through a necrotic mechanism. And so these pathways are highly integrated and highly linked. So what we found, and what my lab has been studying over the past several years, are the mitochondrial regulated pathways. It's become so precise that where people were studying backs and back, these are members of the BCL2 gene family, it's gotten so precise that people can actually determine now that an apoptotic kill typically involves permeabilization changes to the outer mitochondrial membrane, whereas a necrotic kill involves permeabilization changes to the inner mitochondrial membrane where respiration takes place. So that's how definitive the field has evolved. And ultimately, I've shown here in this star diagram, this is supposed to be the apoptosome, and apoptogenic factors like cytochrome C and SMAC can be released from the mitochondria, and they too lead to an apoptotic cell death. But more importantly, this receptor pathway is linked to the mitochondria as well through proteins called RIP1 and RIP3, receptor interacting protein 1 and 3. And so they can directly target the mitochondria. And so the mechanism for this is really still not well understood. But the point is that these protein pathways are highly integrated, and they talk to each other, and that they're genetically regulated, which is quite exciting. Okay, so this is the BCL2 gene family, and the BCL2 family was identified several years ago by Carlos Crochet, Stan Korzemeyer, and John Reed. And they were, again, identifying in B-cell lymphomas translocation breakpoint mutation between chromosome 14 and 8 that showed that these B cells were absolutely resistant to etoposide and cisplatin and other types of chemotherapeutics. And then they realized that, in fact, all the cells in our bodies contain this BCL2 gene family. There are a group of family members that promote cell survival. These are the guys up here. And there are some intermediate proteins that promote cell death, sensors of cell death, and then there are effectors of cell death shown here. And so I'll just take you through this very quickly. The common feature to all of these proteins is a carboxy-terminus transmembrane domain shown here in green. They've got these BCL homology domains, 3, 1, and 2 respectively. And then the anti-apoptotic or the anti-death proteins are the BH4s. They contain a BH4 domain. It's an alpha helical, allophalic domain in the N-terminus. The interesting thing is that these proteins hetero and homodimerize with each other, and they're activated under different conditions. And the protein that I'm going to talk about is BNIP3, and why we got excited about BNIP3 is that we cloned it as a BCL2 interacting protein. So my lab was one of the very first labs to show that BCL2 was anti-death in the heart. So this was done maybe 2000, I think, was the very first publication that showed this. So it wasn't that long ago. And it was very simple. Overexpress this BCL2, this anti-death protein, and ask the question, could it make cardiac cells more resistant to hypoxic injury? And the answer was yes. So the question is, how does that happen? How does BCL2 function? Remember, we knew nothing about the mitochondria. We knew nothing about some of the other pathways. So in a 2-hybrid screen, we were able to show that basically BCL2 interacted with this protein BNIP3. So basically, this is what I had just mentioned, and in fact, it was proof of concept that you could genetically modify a cell, a cardiac cell, which would otherwise be sensitive to cell death by ischemic or hypoxic pathways, and make it resistant by adding this particular protein. So the graduate student in the lab basically said, how does this work? So in a 2-hybrid screen, we went after a number of different proteins that interacted with BCL2, and so without getting into a lot of the detail, we published this extensively over the years, and we came up with this protein called BNIP3. And BNIP3 stands for BCL2 19 kilodalton interacting protein. It's a highly conserved protein all the way back into periphery from sponges all the way up to humans. And so it's expressed at different levels, it's turned on at different times, but our interest in this particular protein is that it's highly expressed in hypoxic hearts. So I'll talk a little bit about that. So really, first up in identifying how BCL2 works in cardiac muscle, we found that it interacts with this BNIP protein. protein. It was originally found in adenovirus. Adenovirus has anti-apoptotic proteins as well. The E1B protein, 19-kilodalton protein, is homologous to BCL2. And so the significance of this really was important to us because it suggested that the interaction between BCL2 and NIP might be an anti-apoptotic pathway that we could therapeutically target. But this slide in particular was the killer. So this slide I show in all the BNIP3 talks, and this is the killer slide. Early days, when people were looking at cell death signaling mechanisms, they would look at the usual suspects. So there was BAX, which is a killer, BAC, etc. And when you looked by Western blot, you could see that these are normal cardiac cells, and these are cells that were made hypoxic. You can see that by Western blot, there's really no change in the expression of any of these proteins, nor is there a reduction in BCL2. And then the myosin is just to show equal loading of the Western. But what was very interesting to us was that, we have earlier time points, the only BCL2 protein that was activated under hypoxic conditions was BNIP3. So the early days, this is a 24-hour Western, but the Western, the protein comes up much earlier, or so does the message, and this coincided with maximal cell death, and that's why we chose the 24-hour time point. But when you look at BNIP3, it's interesting because it's got a transmembrane domain for anchoring both mitochondria and perinuclear sites. It's got a BH3-like domain. It's not identical to BCL2, but it's got this PES sequence sitting in the N-terminus, so it's rapidly degraded. So it's turned over quite quickly, but under hypoxic conditions, it's stabilized. And so over the years, we were curious of how BNIP3 would work, and is it possible that we could use this as a biodiagnostic marker. So from the patients that I've been showing, or the early slide, we asked the question, is BNIP3 released in the blood or in the serum, and can we actually identify it? And sure enough, one of my colleagues collected blood, and we looked pre and post, and there's an induction of BNIP3, and we're trying to develop this as an assay to determine if in fact individuals with good reperfusion predict cardiac function outward. So, even though I showed you that lady who had great perfusion, her cardiac function was quite poor. Does BNIP3 play a role in regulating cell death and cardiac function? So this is what BNIP3 does. It sits basically on the surface of the cells, predominantly the mitochondria, and it's basically it's basically sitting there in a non-integrated manner. But under hypoxic conditions, not only is it activated transcriptionally, it actually inserts itself into the mitochondria. And by doing this, it disrupts mitochondrial function and ultimately leads to the disruption of the mitochondrial function and ultimately cell death. So when we asked the question, does BNIP3 kill cardiac muscle cells? Remember, we didn't know anything about BNIP3 and how it worked. And so just to show you what BNIP3 does and how it kills cells, and I'll argue later, this afternoon, that it causes both a phenotype that's both apoptotic and necrotic. And that's why you have to be careful in how you analyze your data set. So this is classical apoptotic killing. Punctated, fragmented nuclei. This is overexpression of NIP. This is Hirsch staining. Control, overexpression of NIP. And we use a variety of dyes and techniques in the lab to look at something called permeability transition pore opening. These are pores that open on the inner mitochondrial membrane, and ultimately, when this happens, the mitochondrial ultimately die out, and ultimately the cell will die out. So the way this dye works is it sits in the mitochondria. When it leaches out of the mitochondria, it means the pore is open. Very simple. And then there's a viability assay. Very simple. Green cells are living, dead cells are red. So I can see that there's punctate fragmented nuclei. The permeability transition pore opens because the green signal is lost, and the cells are dying. Okay, perfect. That's very nice. One of the very first indications that we knew NIP was involved, we saw it was activated during hypoxia, but we really didn't understand what it was doing. If we simply prevented NIP from targeting the mitochondria by lopping off the transmembrane domain, we not only prevented the nuclear defects, we kept the pore in the closed configuration, so the mitochondria were happy, and the cell death was somewhat decreased. But more importantly, when you look by electron microscopy, we can see that this is intact neonatal cardiac muscle cell tissue. But what's really fun is that when you look at cells overexpressing NIP, you see a variety of things. So you can see evidence of perhaps mitochondrial fission, or is this fusion? Is this autophagy, or is this necrosis? You see swelling of the ER and some of the other spaces. So, Tami and I were kind of joking the other day. There's a lot of things going on with BNIP3, and so what might be happening at Jane and Finch, in Toronto, doesn't necessarily mean same things are happening in Guelph. And so the cell has developed a couple different ways to sort of activate different pathways under different conditions. And so what's cool here is that NIP by integrating into the mitochondria, can trigger mitophagy, a process by which mitochondria are cleared under disease conditions. It can also cause necrosis and apoptosis. And it doesn't necessarily have to be sequential, but the protein has this ability of disrupting the inner mitochondrial membrane, which I hope I will convince you of in the next few slides. So this is total oxygen consumption rate, OCR, taken by using a seahorse, of cells overexpressing NIP. And we can see when you look at normal cells, normal respiration is in blue, and you can see that when they overexpress NIP, mitochondrial respiration goes down. And this is consistent with it, BNIP3 inducing a mitochondrial defect now on the inner mitochondrial membrane. And so we had developed a variety of different tools, and we proved that mitochondrial function was impaired at the cellular level, and we knew that mitochondrial membrane potential was going down. And so we knew that NIP was likely a target of the mitochondria were likely a target of NIP. And so we proved that in vitro, but the question was, does it really make any clinical difference? And so in collaboration with a colleague of mine, Gerald Dorn at University of Washington, we built a variety of mice. These are Crelox mice that we built that were tamoxifen inducible, both direct transgenic and cardiac restricted knockouts. And so we asked the question, what happens to cardiac function when you overexpress NIP or you take NIP away? So the in vitro stuff led us to the in vivo data. And so here's the overexpression. This was published in JCI a few years ago. The overexpressing mice BNIP3. And you can see over a period of time they develop this dilated cardiac myopathy. They impair cardiac function is impaired, and the apoptotic index at this point is increased, commensurate with the amount of cardiac impairment. And more importantly, if we do the converse experiment, we basically knock out NIP, and we subject the hearts to MI. These are wild type and these are mutant, and this is looking over time. This is with gadolinium enhanced MR. And you can see that the guys or the mice that were basically infarcted have better recovery in terms of left ventricular diastolic function and systolic function. And of course, the apoptotic index is absolutely decreased at 3 weeks. But interestingly enough, when you look at the scar and you look at the BNIP3 knockout mice, they still develop a scar, they still infarct, but it's much less, and the amount of scar deposition is far less than the wild type. But there's still cell death ongoing, but the extent of cell death is much less. So the conclusion here was the fact that BNIP3 is playing a role both in vitro at the mitochondrial level. Overexpress it, it kills, and you knock it out, it basically prevents cardiac function from diminishing after infarction. So the model we came up with was very simple. Ischemic injury activates NIP, NIP targets the mitochondria will crap out, and ultimately the cell will die. Very simple, very linear. It's kind of how a grant application was written, because the reviewers, I think, and being one of them, I can say this, sometimes lose their attention span if you write something that's too complicated. So I like to keep things simple, and this was the simple model, naively, that we had come up with. So the first summary of the talk is really that BNIP3 is the only BCL2 gene family member that's activated during ischemic or hypoxic conditions. It integrates into mitochondrial membranes during ischemic or hypoxic events. And basically it messes up the mitochondria, resulting in diminished mitochondrial respiration and ultimately leading to the demise of the cell. And so we believe that targeting BNIP3 under ischemic conditions or myocardial infarction-rich conditions might be an important therapeutic for abrogating cardiac dysfunction and cell death. So what I've tried to do is set the stage and given you maybe 15 years worth of information in the past 20 or so minutes to try to prove that BNIP3 is a viable target to go after clinically. Okay. So that backstory led us to this current story, which I'm quite excited about. And work from here was recently published in PNAS. It came out a little while ago. And I understand that the journal club that you ran involved some of our work on doxorubicin and BNIP3. So for those of you who are unfamiliar with this chemotherapeutic and why I'm going in this direction is that most cancers, particularly lymphomas, non-Hodgkin lymphoma, breast, lung cancers, are typically treated with a drug called an anthracycline called doxorubicin. There are derivatives called daunorubicin, and they all work in a very similar manner. They block DNA proliferation or DNA synthesis and ultimately halt the cancer and prevent it from obviously growing or developing any further. Highly effective. These cancer agents, anti-cancer agents, work really, really well. But one of the off-target effects or one of the side effects of these drugs is that they induce a cardiomyopathy. And so there's an acute cardiomyopathy, I think Kim will agree, and there's a chronic cardiomyopathy. And the acute cardiac dysfunction that occurs actually causes heart failure. And so individuals taking this drug at an early age will develop a heart failure if not closely monitored. And now there's a new form of cardiology called cardio-oncology, where most cancer patients, in fact all of them, are met with a cardiologist. So you have both an oncologist and a cardiologist working together to titrate the drugs accordingly. So if the cancer doesn't kill the individual, the cancer drug will ultimately, because it's messing up the heart. And the sad part about these particular agents is that they manifest the cardiomyopathy later on in life. So if someone had the drug, in their 40s, they may develop a cardiomyopathy later on in their 60s or older. So it's a very prevalent disease. It's a very important form of cardiac dysfunction, and it's getting more prevalent with new diagnostics and people being on these regimes more frequently. Okay, so here's where the science and the fun begins. So again, a picture's worth 1,000 words. And so in some analysis of some of the work that we had been doing and some of our colleagues, we found that the cardiomyopathy that we observed in individuals treated with doxorubicin was really reminiscent of the cardiomyopathy and the mitochondrial injury we saw in cells that were made hypoxic or overexpressing BNP3. So, of course, our world surrounded BNP3, and we asked the question, is BNP3 somehow involved in doxorubicin cardiomyopathy? So again, these are mice taken from wild type and treated with dox. And you can see that it's interesting that the mortality increases dramatically with these mice. And more importantly, morphologically, you can see that the mitochondria are fragmented. There's evidence of vacuolation, evidence of membrane swelling, and essentially the oxygen consumption capacity is impaired. Very much like I had shown earlier with BNP3 overexpression. And more importantly, there was an induction of LDH. So this was indicative of classical necrotic killing. So LDH was increased, mitochondrial function was impaired, and more importantly, a really nice induction of BNP3. So this led us to believe that BNP3 was somehow involved in the cardiomyopathy induced by dox. But let me see if that's true. So again, these are neonatal cardiac muscle cells. It's a viability assay. Living cells are green, dead cells are red, and control cells are quite happy as you increase the dosage of doxorubicin. And these are matched to the clinical correlate of what an individual would get. You can see that there's a dramatic increase in cell death and a correlation with a decrease in total oxygen consumption rate and reserve capacity. So this is what's happening with cells treated with dox. I showed this on the previous slide that there's a really nice induction of BNP3. The seahorse data that basically demonstrates the decline in oxygen consumption capacity. So we're just showing the same data twice. But more importantly, BNP3, which I mentioned, predominantly lives in the mitochondria, and that the cytoplasm is not only induced in cells treated with dox, it finds its way to the mitochondria. So this is a mitochondrial fraction, cytoplasmic fraction, the adenine nucleotide translocase to prove that the mitochondrial fractions were clean. You don't see it in the cytoplasm, and GAPDH, which is predominantly cytoplasmic. Part of cytoplasmic is not in the mitochondria, but it's in the cytoplasm. So this really proved to us that not only NIP is increased, but it's basically in the mitochondria. So permeability transition pore opening occurs with dox. ROS production, this is dihydroethidine now, increases with doxorubicin. And mitochondrial membrane potential, tetramethylrhodamine, declines with doxorubicin. And so then again, it's consistent with a mitochondrial injury associated with doxorubicin. Now this is kind of an interesting slide because what we wanted to know was mitochondrial fission and fusion events. And so if you look at the far, let's see if I can play this movie. So this is mitochondrial dynamics. Are you playing? Go. You don't want to play for me? That's not fun. What you were going to see here are mitochondrial fission events that basically showed that the mitochondria were dancing around and basically fizzing and fusing. Whereas in this particular slide, which won't play, but that's okay, you can see that these were treated with doxorubicin. The mitochondria are hypercontracted and the mitochondrial network actually collapses. I'm surprised it doesn't want to play because it played before, but that's typical of live TV. One of the things that we were curious about, because the mitochondria were somehow involved, and we know that NIP integrated into the mitochondria, we basically wanted to look at the inner mitochondrial function. And this was a really interesting observation for us. So just to remind folks, the various complexes within the mitochondria. There's complex 1, 2, which is part of the succinate dehydrogenase pathway, complex 3, complex 4, the ATP synthetase, which generates ATP, the F1FO ATPase, which is often complex 5, and of course these uncoupling proteins, uncoupling protein 3. And so the electrons, through various reducing equivalents, NAD and FAD respectively, jump from the different cytochrome complex units and ultimately generate ATP. And so ROS production can occur here. This is a major site of respiration. And importantly, uncoupling proteins dissociate mitochondrial ATP synthesis from electron transport. So they act as stop gaps, but under most stop gaps to prevent ROS production. But under basal conditions, we found that UCP3 interacts with cytochrome C oxidase, subunit 1 of complex 4. So there are 13 subunits of complex 4. Here's UCP3, and here's this large complex of subunit 4. And why this became really interesting, and why is he showing us inner mitochondrial membrane components when you're talking cell death, is the fact that we've decided, we actually found that there must be an impaired respiration, and why. And when cells were treated with doxorubicin, there was an impaired complex formation between uncoupling protein 3 and cytochrome C oxidase subunit 1. Okay, how does this work? Let me just show you this. Here's an immunoprecipitation with uncoupling protein 3 and cytochrome oxidase subunit 1. Here you see the interaction under basal conditions, but under conditions in which the hearts or cells had been treated with dox, the interaction decreases. That's this here model. This model shows that the two proteins don't interact, and then respiration is impaired. In all the previous slides I showed, mitochondrial function was impaired. Respiration was down, ROS production was increased, and permeability transition pore was opened. In the following slide, the right Western just shows basically the proteins were expressed equivalent levels. Here's the induction of NIP. And importantly, we see the slight induction of COX-1, the cytochrome oxidase subunit 1, despite the fact that this protein is increased. Where's the mouse? Despite the fact that the protein is increased, the interaction with UCP3 is decreased. So the interaction somehow is critical for respiration. So we asked the question, based upon some of the earlier work, is it possible that NIP is disrupting the complex between COX-1, cytochrome oxidase subunit 1, and UCP3? This would ultimately impair respiration. So the question was, look for NIP expression, see if this complex is disrupted, and then see if you can do an experiment where you prevent NIP from integrating into the membrane, the mitochondria, if you could restore the complex and restore respiration. Let's see if that works. So here's the Western again, another Western. It's not the same Western. Here's the interaction with COX and UCP3. It disappears with doxorubicin. It disappears when you overexpress BNP3, which is the surrogate of doxorubicin overexpression. And now this is a cool experiment. The cool experiment is simply removing the transmembrane domain of NIP and preventing it from integrating it into the mitochondria restores the complex. And on the previous data, I showed that NIP itself can disrupt mitochondrial membrane function, and by preventing it from integrating, you restore respiration. And again, the lysate to prove that all the proteins are expressed at comparable levels, so the lack of effect is not due to differences in protein expression or any sort of spurious effects with the TM mutant. And again, just to show that wild-type NIP is in the mitochondria, the transmembrane domain is not in the mitochondria, it's sitting in the cytoplasm, and again, the various internal controls to show that the proteins are expressed to comparable levels. So that's very nice. What's the physiology? So the physiology is, here's ROS production in control cells. You don't see it. Membrane potential is intact, great. Treat the cells with doxorubicin, ROS production increases, membrane potential goes down, and now we knock down NIP. So this is an shRNA directed against NIP, and you can see that cells treated with dox in cells that do not have NIP do not have ROS production, and their TMRM is preserved. So this really put NIP in the pathway. And just to prove that the shRNA work, here's the induction of NIP in cells treated with dox, and here's the knockdown. So what I'm showing you here is basically proof that BNP3, in fact, was knocked down. And then this is the quantitative data for ROS production and TMRM. So the data I'm showing is building a case to prove that doxorubicin not only activates NIP, it's causing a mitochondrial injury, and it's BNP3 dependent. One of the things that we were also interested in was mitochondrial permeability transition pore, and this is typically activated by calcium. And so here's mitochondrial calcium content. It goes up with doxorubicin. If you knock down NIP with shRNA, you can prevent mitochondrial loading of calcium and also to another approach. We use the TM mutation that prevents NIP from targeting the mitochondria. If you don't like shRNA, or still not convinced with my shRNA experiment, here's another technique where we prevent NIP from targeting the mitochondria, and we also prevent calcium overload in the mitochondria, concordant with preserved mitochondrial permeability transition, so it doesn't open. Here's the pore opening with doxorubicin, and when you knock down NIP, pore stays in the closed configuration. So basically, the pore opening is a calcium-dependent phenomenon. What about cell survival? Okay, so here's living cells. The cells are green. They're quite happy. You treat them with dox, they die. Here's the quantitative data for LDH release and troponin. But more importantly, you take away NIP from integrating into the mitochondria. By using the TM mutation or shRNA, you virtually rescue cell death from occurring in cells treated with dox. And more importantly, I showed on the very first slide an increase in LDH release, which is indicative of necrosis. Here's the necrotic kill with doxorubicin. It's knocked down with BNP3, shRNA, and similarly the troponin T, which is another marker of necrotic cell death. High sensitivity troponin is also impaired release when you knock down NIP. So what I'm trying to build the case is that the intermitochondrial membrane defect induced in cells treated with dox is mediated by BNP3, and it results in necrosis. Whoops. Let's continue on. Okay, so I showed all this in vitro. What about in vivo? So let's go back to the in vivo mice and the in vivo story. So here's saline-treated wild-type mice treated with doxorubicin. Again, you can see the nuclear, you don't really see the nucleus here, pardon me, mitochondrial defects, vacuolation, and disrupted mitochondria. You see an increase in LDH release in cells treated with dox. If you take a look at the BNP3 knockouts, the mitochondria look they're pretty much intact. The mitochondrial sarcomeres, I hope Glenn likes this image, are intact and they look pretty okay. And even when you treat them with doxorubicin, the mitochondrial architecture looks pretty good, and the myocardial filaments remain intact. So this is kind of interesting because when you also treat the BNP3 knockouts with dox, you abrogate the loss of LDH release. So there's less LDH release in the BNP3 knockouts. One of the reviewers asked us, and it's a good housekeeping control, we didn't otherwise do it, at least included in the paper. They wanted to know if BNP3 is activated in the BNP3 knockouts. I guess it was a reasonable question to ask. I wasn't going to argue. I mean, okay, I'll do the experiment. It's a knockout. What do you expect to be expressed? So anyway, here's the induction of NIP message in wild-type mice treated with doxorubicin, and here is the lack of induction of BNP3 in the BNP3 knockout. So anyway, I did this experiment just because the reviewer asked. But anyway, just to prove that BNP3 is not expressed in the BNP3 knockouts. Cardiac function, wild-type mice treated with doxorubicin, cardiac dilatation occurs. These mice ultimately go into failure. Ejection fraction is impaired. More importantly, when you treat BNP3 knockouts with doxorubicin in contrast, you can see that cardiac function is ultimately preserved. And cardiac function between the knockouts under basal conditions is very similar to the wild type, so there's no failure. And you can see longevity. Even the BNP3 knockout mice, while they outlive the wild-type mice, there's some cell death and some cardiac failure occurring. And these numbers approach 15 in some cases and 10 in others. So it's a good number of cells and hearts that we treated. So one thing we were curious about was mitochondrial function. So we extracted the mitochondria from the intact tissue, and we repeated what we did in vitro, and we asked the question, is mitochondrial respiration rescued in the BNP3 knockouts? And again, this is just to show you the seahorse data for the wild type treated with dox and the BNP3 wild type and knockouts treated with doxorubicin. And I'll just draw your attention to oxygen consumption rates. You can see that the decline occurs in the wild type mice. And you can see that that decline in mitochondrial oxygen consumption is preserved in hearts or mice treated with doxorubicin in the BNP3 knockouts. And then again, the reserve capacity is completely protected in the BNP3 knockout mice. And again, this was unbelievable because it absolutely reproduced what we saw in vitro. Again, these were mitochondria isolated from the BNP3 and wild-type mice. So this is the model that we came up with. The doxorubicin basically will transcriptionally activate NIP. We have ongoing studies to look at how NIP is activated under basal conditions, under hypoxic conditions, and now with doxorubicin. We believe that the activation of NIP allows NIP to integrate into the mitochondria. It disrupts the inner mitochondrial membrane, resulting in necrosis. And this is largely due to disrupting of this complex between UCP3 and coupling protein 3 and cytochrome oxidase subunit 1 on the inner mitochondrial membrane. ultimately leading to an increase in ROS production, so radicals increase, mitochondrial membrane potential declines, and the cells die. And what I hope I impressed upon you in the past 40 or so minutes was that I show by not one, but by 3 independent approaches that if you abrogate the ability of the cells to signal through BNIP3 using the BNIP3 knockout mice, and in vitro the TM domain mutation that can't integrate into the mitochondria, or by knocking down BNIP3, we completely preserve mitochondrial function, and you prevent cell death from occurring, and you promote cell survival. So this is something that we've been engaged in, and this is work that we've started, our collaborations with our folks, stereostructural chemist at Albert Einstein University, Everest Gavotheosis, who has kindly helped us build these peptides that we're going to target the TM domain of BNIP3 and build therapeutics to actually target BNIP3 integration into the mitochondria in a preclinical test. So we've developed the peptides. We're testing them in models of ischemia reperfusion injury in large pig models with my colleagues, and we hope to see how this will work as a therapeutic in preventing cardiac cell death and predominantly necrotic cell death post-myocardial infarction. So to summarize and conclude, BNIP3 is activated in cardiac myocytes or hearts treated with doxorubicin. We believe that the mode by which BNIP3 causes cardiac cell death to occur is by disrupting the inner mitochondrial membrane, by disrupting complexes between UCP3 and cytochrome oxidase subunit 1 of complex 4. This is really one of the first studies or stories, if you will, that link inner mitochondrial membrane defects to BNIP3 and necrotic cell death induced by doxorubicin. And so this is very paramount because, in fact, doxorubicin had been shown to cause necrotic cell death, but the downstream effectors were poorly understood. And so we believe that targeted interventions that abrogate the ability of NIP to integrate into the mitochondria in individuals that are undergoing ischemia reperfusion injury or treated with doxorubicin will have a major impact on preserving cardiac function, particularly in cancer patients treated with these toxic agents. So I'd like to thank the funding bodies, CIHR, Heart and Stroke, the Canada Chair Program, my collaborators past and present for their wonderful insight, guidance, reagents. It's just been terrific. And of course my fabulous crew who are here. Some have graduated, some are present, who have done a majority of the science and allows me to stand here and present this work to you. So with that, I'll stop and be happy to take questions. Again, thank you, Tami, so much for the very kind invitation to be here. I'm absolutely delighted. And, congratulations to you all for hosting such a wonderful cardiovascular event. So thank you again. So the answer is no. True, and unrelated. So it's a great question, and I'll tell you why. So we pulled out the NIP promoter, and we found NF-kappa B elements in the proximal promoter. There are 2 NF-kappa B elements. We published a couple papers in Circulation Research and Circulation showing that the NF-kappa B, the P65 subunit of NF-kappa B, actually recruits a histone deacetylase, HDAC1, and acts as a transcriptional repressor under basal conditions. During hypoxia, this complex, this inhibitory complex, is released. There's a dissociation between the NF-kappa B and the HDAC, and so the NIP gene is actually activated. Even if you activate HIF, if this complex is sitting on the promoter of BNIP3, this NF-kappa B HDAC complex, you won't drive NIP expression. Under conditions where, under these conditions with doxorubicin, it's a HIF-independent mechanism, but it's dependent upon the dissociation of this inhibitory complex. So I didn't get into it because, it'd be complicated, but it's a great question, so thank you.
But just a follow-up
Yeah.
to it then, functionally, does it mean maybe that the cell is thinking it's hypoxic, but it's not, because BNIP is part of a classic hypoxic response?
That's correct.
But you've got lots of potential oxygen around, so it drives malignant, for lack of a better term, maladaptive responses.
Exactly. And so what I think, and some of the things that we've been tossing around in some collaborations with others and Tami, were the fact that the mitochondrial death, or the deaths of selective mitochondria, are important. So again, the analogy, and I hope you don't live around Jane and Finch, but I love that analogy, because you get rid of some of the things that are going on over there, you don't affect the rest of the city. And so the activation of NIP selectively may allow the cell to dial up or dial down the number of mitochondria that might ultimately be contributing to the demise of the cell. So you may have 1,000 mitochondria, you cut it down to 800 or 700. This is just my theory, and our data somehow support this, that allow the cell to still survive while it generates new mitochondria as the cell lives on. But if you wipe out the entire mass of mitochondria, the cell will ultimately die. So yes, you're right. It may view itself as in a hypoxic status. It may think it's hypoxic, even though there's enough oxygen present. Thank you.
Similar sort of comment. It was a real tour de force. Really enjoyed your talk. I was interested in the protein accumulation of BNEP3 and the relative roles played by what sounds like transcriptional de-repression relative to the insertion in the mitochondrial membrane. And you highlighted for us the PES sequences that were there and indicated that there was probably very rapid protein turnover. So I was at first was grappling with the fact that how does transcriptional de-repression or an increase in transcription actually drive an increase in the protein when you've got PES sequences that should drive its
degradation.
Yeah,
Absolutely. Again, thank you. Wonderful question. So the way I think, and it's again my view of the world. This protein is toxic. It would kill cells, particularly cardiac cells. And so we think that the protein is rapidly turned over. The message is there. You can see it under basal conditions. You can see the protein under basal conditions. But it depends on how you look. You basically have to use lactocysteine in some cases to block the proteasome in order to see a lot of NIP accumulation. I think many things are going on during hypoxia. Let's use hypoxia as an example. The proteasome is inhibited, so that contributes to protein accumulation. We've mutated the PES sequences by directly mutating. There's there's ways we've done it, but importantly, chopped off the N-terminus. And again, you change the characteristic the way the protein integrates or functions. So there are many things going on, and this protein is so highly regulated. It's phosphorylated, it's regulated at the transcriptional level and at the protein level. And so one of the things I didn't talk about because of the time and because I could be here all afternoon, we found a splice variant of BNIP3 that's actually pro-survival. And this protein actually binds to and inactivates NIP. And you never see it expressed under basal conditions. It's got the same N-terminus of NIP and is rapidly turned over. But under hypoxic conditions or ischemic conditions, we see the accumulation of this protein, and it actually binds to and tethers BNIP3 from targeting the mitochondria. So times where the cell may want to dial up or dial down the number of mitochondria it wants to get rid of, it regulates its mitochondrial pool by regulating the splice variant. And I think that's the key of how NIP is regulated. So the translational regulation, post-translational regulation is probably key through this splice variant. So if I'm invited back again, maybe next time I'll have more information to share with you. So, but thanks for that great question. Glenn.
So Dr. Rubinson is metabolized very differently in mice than it is in people. I was just wondering if you had any samples from human patients to show the pathways were similar between the two, or if there's a metabolite that's produced that's unique.
Yeah, that's another interesting question. So we have some ongoing collaborations with our clinical colleagues to actually look at that. We can detect blood, BNIP3 full length in the serum of individuals that are undergoing reperfusion or stenting, or PCI. So I think that's something we're going to do with Dr. Rubinson. And now we're just in the process of collecting tissues. And we have a whole clinical cohort looking at that. So we've got echo, blood work, cardiac function, and that's something we're curious about. But absolutely, because we'd like to use BNIP3 as a diagnostic marker for cardiac function in these individuals, in addition to abrogating its ability to kill.
Thank you very much.
Thank you.